Professor of Pediatrics, Computer Science and Engineering and Bioengineering at UC San Diego, where I direct our Center for Microbiome Innovation. In my main talk, I told you a lot about the microbiome, but not about how we measure it. And I wanted to share with you a little about the techniques we use to find out about the microbiome and why the concept of a microbiome map and where you're on it is really important in the research we do. The way we look at the microbiome is typically by reading out the DNA of the microbes. And there are two ways that we can do this. One is by what's called 16S ribosomal RNA profiling, where we take one gene that's kind of like a name tag for the bacteria, and we make billions of copies of it by a technique called the polymerase chain reaction, or PCR, and then we can read that out by DNA sequencing. The second technique we use is called shotgun metagenomics, where instead of looking at one particular part of the genome that's a very good marker, we smash the whole genome up into little pieces, and then we sequence each of those pieces and try to puzzle them back together in like an incredibly complex jigsaw puzzle, where what we're looking at is billions of pieces spanning thousands of different species. The results of both of these are essentially a table where for each sample, what we know about is we know what kinds of microbe were in it and what the relative abundance of each of those kinds of microbes were, which we read out by the number of pieces of DNA we saw that belong to each of those microbes. And these tables are incredibly complex, especially when you're trying to compare a lot of different ones. To put it in perspective how complex it is, each teaspoon of stool that you produce has about as much data in it as stored in the microbial DNA as it would take literally a ton of DVDs to hold. So you can think about that the next time you're taking a data dump, as it were. So we have this huge problem with data in the microbiome, and what we have to do to get around that problem is we need better ways of taking the species profiles or the functional profiles that tell you what all of those species do, and making some sense out of them. And I have to tell you that, especially now that this is more and more common from a clinical perspective, it's your doctor's nightmare that you're going to get your microbiome profiled through one of the citizen science projects like American Gut or Microzetta, or one of the companies that does microbiome profiling now, and show up in his or her office and say, "Hey doc, I have some great news for you, which is that I had my microbiome done, and now I have this list of 1,000 species they found in my gut. Or better yet, I had it done with shotgun metagenomics, and I have this list of 2 million genes that they found in my gut. And with all this data, you can tell me what's wrong with me, right, in the 12 beautiful minutes that we have together." And what's your doctor even going to do with that? Refer you to colleagues in psychiatry for being crazy enough to think they could do something with such complex information in such a short time that you have together? So a lot of what my lab tries to do is to make it not crazy to think that you could get something out of that profile by combining it with information from other people and even from animals and from different environmental samples around the planet. And the way we do this is that we need a very precise way of measuring the difference between those profiles, which we do with a distance metric that Kathy Lozapone, then a very talented grad student in my lab and now tenured professor at the University of Colorado at Denver, developed back in 2005, called Unifrac, where we use Darwin's insight that there's one phylogenetic tree that connects all life to look at the locations on that universal tree of the microbes in each sample, and then we can compare two samples based on the distance between the microbes in that tree. And so what's exciting about that is we can use another statistical technique called principal coordinates analysis to distill all of the microbiome information in a sample down to just one point, where 2 points are close together in these displays if they have more similar microbes as read out from their evolutionary history, and then they're further apart if they're more dissimilar in terms of the evolutionary history of those microbes. And so what's exciting about this is that for huge projects like the Human Microbiome Project, where we're looking at thousands of samples and we're looking at billions of sequences, we can take all of that data and turn it into a map, a picture that you can understand. Now we're starting to come to realize how your location on that map is important, and that's what I talked about in the main talk. but also on our journey on that map. How our microbiomes change over time is also really important for health and especially for disease. And my personal journey into discovering this is very much one that I undertook together with Larry Smarr, who's another professor at UC San Diego and is in charge of an institute called CalIT2, so the California Institute for Telecommunications and Information Technology. And Larry's a very interesting character because he's been involved in supercomputing for a long time. He's originally an astrophysicist, and he's very interested in big data and data visualization topics. And shortly after I got to UC San Diego, a little over 5 years ago, I was explaining to him the Human Microbiome Project dataset and how we could visualize it on this huge display wall, so a screen the size of a wall with 65 million pixels. And he was looking at all these points representing individual samples from the feces, the mouth, the skin, and so on of people in the Human Microbiome Project. And he said to me, you know, Rob, I have a very interesting microbiome myself because I'm an inflammatory bowel disease patient. How do I get myself onto that kind of microbiome map? So shortly after that conversation, this box of Larry's stool on dry ice showed up in my lab. And Larry's a very interesting character, not just for that reason, but because he's a leader in the quantified self movement. So he's been measuring things about himself and releasing them on the internet for the last 20 years, starting with one dimension, which was his weight, and then going into dozens of dimensions with blood tests, thousands of dimensions with human genetic profiling, and then millions of dimensions with the microbiome. And had been collecting his own microbiome for a couple of years at the time that I met him, but it was very difficult for him to get any usable patterns out of it, because if you just plot the different kinds of microbes that you have over time, like say Ruminococcus versus Bacteroides versus E. coli, there just wasn't any clear pattern. But when we made a microbiome map for Larry just out of his samples, we saw something very interesting when we connected up his journey over time, going point to point on that map. Because what we saw is that his movement wasn't random. He started very much at one corner of the map and then traveled directionally until he had a change of medication, where he then switched rapidly into a new place on the map, and then in that new place he bounced around more or less at random. And what was exciting about that is that he'd collected very detailed clinical information about himself. And so what we could see is that in that period of directional change, what was going on is that he was losing weight, he was having frequent IBD symptoms, and his life was not very good. When he changed his medications and got into the new region of the map, where he was no longer changing directionally and bouncing around at random in microbiome space, his weight went back up to a healthy set point, and then he stayed there for months afterwards. And had we known about Larry's individual microbiome map and how he could journey through it, we could have told him that as soon as he got into that new region, he was going to be fine for months afterwards. Now, you might think that that's all very nice for someone like Larry, who runs a large institute and has all these cool computational and visualization tools, but could we do it for other people? And so in collaboration with Janet Jensen at Pacific Northwest National Labs and a number of other collaborators, we were able to look at dozens of people who had ileal Crohn's disease, colonic Crohn's disease, or ulcerative colitis, and compared their journeys on the map over time to people who had to people who were healthy. And what we saw was that not just their locations on the map, but how they traveled across the map was totally different, where especially the ileal Crohn's patients moved a lot on the map into a completely different region sometimes, whereas healthy controls stayed very much in one side of the map. And what was amazing was that we could show that by looking at people's movement on the map, not just a snapshot, we could get better disease classification than calprotectin assays, which are currently the gold standard for measuring inflammation. Now, in subsequent work with Larry, we were able to observe some extreme changes in his diet. So he went vegan and gluten-free for a period of several months, and then he went off that diet and onto a diet where, inspired by the American Gut Project, now finding about the number of kinds of plants people eat having a big impact on the microbiome, he decided to drink a smoothie every day that contained at least 40 species of plants. And what was amazing was that the impact of that smoothie had a larger effect on his gut microbiome as measured by the location on that map than being vegan and gluten-free. And so what's fascinating about this is that it shows that more guided interventions can have a major impact on your gut microbiome and on your journeys through the map, and we think on your health. And so our vision here that we're trying to build on is to understand how we can build not just a map, but a kind of microbiome GPS that tells you not just where am I right now, but where do I want to go to get into a better, healthier region of that map, and what do I need to do in terms of my diet to get there, whether it's cutting out a particular ingredient that causes inflammation, even though it's healthy for most people, or whether it's avoiding particular additives, whether it's prebiotics or probiotics, whether it's particular ingredients like all those colored molecules that plants make, like carotenoids, lycopenoids, and so forth, that help shape our microbiome over time. And so what we're hoping is that we can give each person this kind of individualized microbiome GPS. We're making it easy to measure the microbiome day after day after day, rather than just a snapshot that's expensive that you take once. We could help you understand how you're moving around on that map and how to modify your microbiome over a lifetime with diet to preserve health.